Awhi Māori kia koutou katoa, ngā mihi, kia koutou katoa, nau mai haere mai. Welcome to Rangahau Rising. My name is Nick Waipara. I'm your MC facilitator slash chairperson of the day today. Welcome to you all to this kōrero. Before we start today's hui with Sarah, I'll just do a quick karakia to bless our hui today. Tūtaue mai e ranga, tūtaue mai e raro, tūtaue mai e roto, tūtaue mai e waho. Kia tau ai te Māori tū, te Māori ora, te hei Māori ora. So as I said, nau mai haere mai, welcome to today's Rangahau Rising. I'm really excited about today's kōrero. It's going to be really fascinating and we're going to learn a lot. Just some housekeeping for those. We're going to keep the questions to the end and please put them in the chat and we're going to, I'll read out any questions or any points of clarification or any things you'd like to ask Sarah. So please use the chat at the end. But in the meantime, I'm going to present our amazing speaker today, who's Sarah Herbert, is early career researcher at Victoria University of Wellington. Her research is focused on the understanding of the perturbations on populations, communities, and ecosystems through empirical study and statistical modelling. She's completed a postdoctoral fellowship with Bioprotection Aotearoa at the end of 2024, and I'm really looking towards her mahi today. So please welcome Dr. Sarah Herbert and her talk today, Calculating Hope, How Spatial Modelling Can Help Us to Plan Strategies to Protect Aotearoa New Zealand's indigenous myrtles from disease. So kia ora, Sarah, the floor is yours. Welcome.
Oh, tēnā kōrua, everyone. Sorry, tēnā koutou. There's way more than two of you. Yeah, thank you so much for coming along, and I think we'll just get cracking, eh? So, sorry, just 1 minute. I'll just share my screen. Here we go.
Yes, it's loading. It's loading.
Lovely.
Yep, we
Right,
can see it. Yep, there we go. All good on my end anyway.
great, fantastic. So I think Nick's done a really good introduction, so we'll just go straight into it. Firstly, what is a myrtle? I know most people in this audience are going to know what that is, but just in case you don't, and also because I learned a little bit putting the slide together, the Myrtaceae family is a family of plants and it's massive. There's about 5,950 species and 132 genera, and you're most likely to be familiar with several myrtles. So everything from like the cloves that live in your spice drawer to the eucalyptus that are planted around the country to the Pōhutukawa that you see all around cities and the coasts and our beloved feijoas and mānuka. These are all myrtles. Now, this number is a bit debated, but I'm going to just say we have 18 indigenous myrtle species in New Zealand. We can talk about that a bit later. And also, we've got numerous introduced species. I said numerous because I did a quick count a while ago, and I think it's about 30, but there's stuff that keeps coming up in the country, popping up in people's gardens all the time. So, what is myrtle rust? So, myrtle rust is a plant disease, and it only infects plants in the Myrtaceae family. But it's actually really, really unusual. It's this pathogenic rust fungus that has come from South America. And it's really weird because plant diseases often don't have that broad a host range, but this one's been really successful. It's spread to several countries, mostly in the Southern Hemisphere, and it infects at least 480 species of myrtles globally, which is just like the biologist thing is it's when you're not thinking about it being a problem, it's actually quite a fascinating organism in itself. And because of its subtropical origin, it really likes things hot and humid, which is a really salient point that I want you to remember because we're going to talk about that later. I've also popped a link in this slide, in the previous slide, to the wonderful myrtlerust.org.nz website. So if you want to find out more about these things, that's such a good resource. So why does this matter? Well, myrtle rust, it's causing really large-scale population declines of susceptible myrtles, and it's rapidly changing Australian forest communities. So we're sort of unlucky and lucky at the same time that it got into Australia quite a bit earlier than it did New Zealand. So we're able to look at what's happening in Australia and have a think about, how's that going to affect our country? So to compare, myrtle rust got to Australia in 2011. It got to New Zealand in 2017. And while we were still kind of waiting to see what the impacts are going to be on New Zealand's flora, in Australia, the picture's really, really scary. So, they've got a lot of eucalypts and other myrtles in their native forests. And what's happening is we're seeing a lot of dieback, such as in the picture. This is a native stand of mature Rhodomyrtus poseidoides. Most of the trees in that stand have been killed within just 2 to 3 years of myrtle rust getting into that stand. And it's causing a real shift in the composition of the forest communities. And we just don't really know what that's going to mean for all the ecosystem services that these forests provide, let alone the habitats for wildlife. And so, like, our question is like, we've got a really myrtle rich flora in New Zealand, perhaps not as rich as Australia, but, what's going to happen to our myrtles if the disease establishes as an Aotearoa? And I mean, I should also reiterate that this affects commercially important species too. Um, it's got a relatively low impact at the moment, but it can affect mānuka, it can affect feijoas. So why use models? And I just put this quote here because I love it, and I think it's always a really good thing to kind of keep in mind when you're modeling or when you're using the outputs of models is essentially all models are wrong, but some are useful. And the reason for this is because a model to be useful was always going to be a simplification of reality. I think a really good way to think about it, if you think about the way that we understand our world or just other complex systems, we're all walking around with the model of the world inside our heads. But to really understand it, we can't really capture all the component parts because then it gets really messy. So any model was always going to be a necessary simplification of reality. And what we try to do as modelers is just capture the most important parts. So hopefully our models aren't too wrong and hopefully they're useful. But basically, I've been really, really inspired by amazing work that's been done overseas in both the agricultural spaces and also in the forest conservation spaces. There's just really cool stuff, particularly coming out of actually the Gilligan lab at Cambridge University. There's a guy called Ivan Sobronenko who's done all this really cool recent work. He's created these models that basically allow you to forecast what the optimal spatial arrangement of crops are. This is cassava crops based on how is that going to affect disease spread. So it's sort of about like trying to find that sweet spot between like what sort of density should you plant cassava at versus what's too dense and it's just going to like explode with disease. And I just keep seeing this kind of theme again and again and again, even in things like ash dieback. I got really, really inspired by a beautiful study from North America on sudden oak death, where they're able to actually forecast like what factors in the environment affect disease spread, and therefore, should we target certain stands of trees and cull them to manage the disease? Really, it's really funny because it's a really sophisticated way of effectively creating these quite low-tech solutions, such as just like, where do we cull the trees or where do we plant more? But I think that's really cool because we don't really want to be spraying fungicides into our environment all the time. And even when we need to use fungicides, I think we need to be quite smart about, where do we spray? We don't want to be wasting resources. So I think models are great, personally. And the particular models that we use address this issue of where should we do species conservation? Because I'm a pragmatist, ideally I think it should be where our efforts are going to be efficient and most likely to pay off. But really, it's a simple, it's kind of a simple idea, but one that's actually quite complicated because there's so much spatial variation to consider. It's a complex system, and you want to know where is the habitat suitable for your target species that you want to conserve? What's the kind of landscape of risks and opportunities? And thirdly, the thing that often doesn't get considered very well, is actually just the human dimensions, like what sites are actually accessible, what like values do we hold on the land, and how do people interact with the landscape itself to manage? Because ultimately, it's the people doing the management, so we need to make it feasible for them. And we will come back to this in our first case study. So, this was a really interesting paper because this was actually one that got started by the previous postdoc in my position, and then it ended up picking it up and taking it across the line. But it's ended up being, it was a really, really, really good case study to basically test some of these really quite novel methodologies that we were trying to get a really good handle on disease risk and again, this landscape of risks and opportunities and habitat suitability, as I keep talking about. It's been published. The link is just below on the slide, so feel free to check that out. And our target species for this was myrtle, obviously. A beautiful myrtle called Syzygia mairi, or mairi tawake, or swamp mairi, or waiwaka. It's got a lot of names, and that's because it's really, really important, particularly to a lot of the iwi and hapu who lived around these wetland areas that it inhabits. Unfortunately, it is critically endangered, partly because it's a wetland obligatory, and we've been great at draining all of our wetlands. I think the scale of wetland drainage in New Zealand is actually higher than the baseline internationally. So, unfortunately, this tree would have been across a lot of New Zealand, particularly in places like low-lying basins like the Wairarapa. But we've drained them all. So it's had a lot of historical habitat loss. And now with myrtle rust incursion, turns out it's one of the most susceptible species to myrtle rust. So it's got this like awful perfect storm of like interacting threats now. So I think this is what makes it a really important species to focus on because it's possible that there's a risk of functional extinction within less than 10 years if we do nothing. It's pretty scary. So why did we choose Greater Wellington? Not just because our whole team lives here, but it was actually because it's more achievable to survey a region of New Zealand for additional presence records, of which we actually had a real dearth of for Māori Tāwaki. Secondly, at the time, there was some really nice high resolution data available from the Greater Wellington Regional Council that we could use for hydrological modelling, which is really important when you've got a tree that needs a wet landscape because you need to know the hydrology. And turns out our gamble, it did actually pay off. So there's some previous modelling that's been done for this species, and it's awesome work. But I think they were a little bit limited at the time by the methodology, so they had to use the presence absence data from the National Vegetation Survey. And if we look at the records in Wellington, you'll see that there's only 5 of them in the Greater Wellington region that have been recorded, but like we know there's way more than that. And so my colleague, Colin, who recently finished his PhD on Māori Tāwaki, he went and surveyed for them and he found 268 additional locations to add to the 5. So that was great. But we had the challenge of combining 2 really different data sets. Presence absence data normally comes from systematic surveys, whereas presence only data is kind of loose. Like you go and record the observations as you find them. And if you're a numbers geek like I am, then you kind of realize, those are quite different observation processes, and we need to account for that in our model in order to actually have like a good model. So we were actually really lucky, and there was a student from Norway who had recently developed these integrated species distribution models, and they're really cool because they can use systematic records as well as all the citizen science data that comes about as these presence only records, and you can combine them into the same model and seems to, well, I mean, we've found that it actually performs really, really well. So I'm just giving you a bit of an overview of our process, and it's basically you'll see all these things have a lot of steps, which what makes them fun and also kind of annoying to work with sometimes. But basically, we pulled in our spatial records of our Māori Tāwaki. We got a lot of environmental environmental covariates as well as observer bias covariates because, of course, you're going to record more trees in the places you can actually get to. So we use the distance from the nearest road as a covariate to try to correct for that. We pulled these all into an integrated species distribution model, and it spits us out a quantity called Mighty Tawaki point intensity, which just means the estimated number of point locations that you'll get based on the data that you have. Next, we use the land use capability indicator model to effectively model flood concentration across the Greater Wellington region. And we use that as a proxy measure for soil waterlogging. Then we also found another really recent method that had been developed, and that's how to make filters for things that you can't easily put in a species distribution model. So it's actually fairly simple. It's just bootstrapping. So we did that, and we basically came up with a soil water filter, which we were then able to multiply by our point intensity estimates and actually get like a really nice refined picture of where the good habitat for Māori Tawaki is in the Wellington region. Um, this is what it looks like. So these are our unfiltered point intensity. Um, they actually at this point in time we found these models quite hard to interpret. So we ended up kind of looking at the point intensity and how that differed from the regional mean. So what you just need to take away from this is everything that's in blue, green, and yellow is high quality habitat because there's more points than expected than the mean. And everything in purple is low quality habitats because there's less points predicted than the mean. This is what our flood concentration layer looked like. And after we did the bootstrapping, what bootstrapping does is you compare the expected number of points that you get from doing this 10,000 times across the region. You compare that with the observed number of points, and basically it allows you to figure out what habitat is suitable and what's not based on whether you've got the same number of points as expected or if you've got more. So we were able to say, okay, I mean, fairly intuitive areas that don't have a lot of like floodwater concentration are not going to be suitable for Māori Tawaki, which is it's good because we know that it likes wet soils. But you can see we're able to turn this into a soil waterlogging filter. And then when we multiplied it by our filter predictions, you can actually see that it really restricts the area of suitable habitat in the region that Māori Tawaki can grow in. And I think this is actually a really important step because if you compared it to our previous kind of iteration of the model, you'd almost be fooled into thinking you had a bit more land to play with than we actually do. So that was useful. And basically we found that there's 1,230 kilometers squared of suitably waterlogged soils in the region for swamp Māori, which is it's not bad, actually. It's quite a big area. And then we took our model outputs to answer the question, well, out of those areas, what's actually feasible for management? And we looked at 5 scenarios here. So just what can we actually get to? And that was defined as being within 500 meters of a road. Um, we also looked at the areas that have very low myrtle rust risk. You actually see it's quite depressing. It's a really tiny area that has really low myrtle rust risk in Wellington. But if we give it a bit more of a permissive scenario, so just kind of low myrtle rust risk, you can see that we've got a bigger area. And then, of course, we just looked at what land is accessible and has very low myrtle rust risk, and what land is accessible and has low myrtle rust risk. And basically, you can see, like, as you add on each of these filters, the area that you have to play with shrinks. But anyway, that all seems quite abstract. So I wanted to give you a really concrete example of how we could use this. So we put this together for a presentation to Kohanui Marae. And so say you're in an area and you've got all of these wetland sites of interest. These are the pink outlined parts. And you want to know which ones are suitable for Māori Tawaki and what's the level of risk? Like what should we conserve? So you can see here that yeah, as expected, like most of these wetland sites actually like, they're good habitat. So that's great. So you can actually plan within them at a scale of 100 meters by 100 meters. Like, yeah, we should put Māori Tawaki there, but maybe we shouldn't plant it there. However, this area in black is all the areas that are at quite a high risk of myrtle rust. So you can see while the situation might look a bit dire, you actually can identify these really interesting little opportunities, such as the north part of Wairoa wetlands here, or along the shores of Lake Wairarapa, that actually seem to have lower myrtle rust risk and might be some really good sites to plant more Māori tawaki. I should also mention these GIS layers are now freely available to download, and currently working on trying to get some interactive maps put up on the web so that you can just literally go in, look at your site, and see how much risk is there and is this good for Māori tawaki. We then asked the question of, well, how much of this land is actually manageable? And basically, the more restrictive your scenarios get, like the better the area is for Māori tawaki, you get less and less land. And then we kind of wanted to know, well, how many trees might be in those sites that, have a pretty good prognosis under this myrtle rust pandemic scenario? Well, we predicted across the region that we'd have Māori tawaki at 712 locations. But out of this, if we were able to manage all of the suitable habitats, we would get, so I actually can't see the number here. It's about, yeah, we would get about 610 of those sites would be captured. And I think it's less because there's just some error in that model, but you do a pretty good job of protecting most of the Māori tawaki. But if you were only able to protect those sites that had low infection risk, that number drops to 472. And then if you were to only, if the ones in the very low infection risk sites are going to be okay, the number drops to 46. But then if you can only protect the trees that are inaccessible sites, then it drops to 43. So what this means is that protecting locations that have existing trees in only the lower risk sites or accessible sites, it's not going to maintain current abundance if myrtle rust indeed does cause declines in the Wellington region, which sounds like a really dire scenario. But I think what we learned from this is that I think actually some good work is achievable as long as we're just adapting our actions to the local conditions. And I think that's where the models are quite helpful because it allows you to figure out what's the conditions in my area and what do we need to do. So, for example, in the habitats that are less accessible and but have like quite a high infection risk, we probably should just be prioritizing collecting seeds and cuttings for propagation because we've got the highest risk of losing those trees. Whereas in the area, it's quite a small area, but it exists. We've got an area that has lower infection risk and is accessible. So maybe we should be like planting and like really intensively managing the trees in these areas because we're likely to have the best outcomes there. So I think it's not all dire. I think it just you know, I think we just, need a use for these models and we just need to plan and coordinate actions and again respond to the local conditions. And now that brings us on to something kind of different, but actually very related. This is our second case study. We've called it MIRDI, stands for Myrtle Rust Disease Intensity Index. And we developed it to actually try to figure out, well, what's the impact of exotic myrtles on disease risk? And it's because of things like this happening. So this is in my local Mitre 10 garden centre. This is Syzygium austral. It is from Australia and it gets used a lot in hedging and it is highly, highly susceptible to myrtle rust. And it was actually really funny because when Rebecca and I my colleague Rebecca and I we've been looking at the myrtle rust records in iNaturalist and we were like, oh, you can just see these like lines of records and we realized we were like, it's lily pilly hedges. So we kind of think that we keep joking that myrtle rust disease intensity is probably correlated with like affluent suburbs where they plant a lot of lily pilly hedges. And this is an observation that's come from the Auckland region, and we really wanted to see is that a national thing or is this an Auckland thing? So again, I'm going to give you a lovely flow chart because I like these. Basically, it's actually quite similar to what we did with the Greater Wellington work, except just vastly expanded. And we also got I ended up collaborating with the guy who actually developed the integrated SDM models in the R package for that. And We've just refined our processes a lot better. So now we still use integrated species distribution um, models, but this time we were able to kind of get an index of the actual density by doing a really fancy thing where you just take the integral of your fitted function. Something I'm happy to talk about at length, but I'm sure no one else wants to. Um, so we did that. And then again, we actually did this filtering thing again, except this time we used it to look at land cover because we were like, well, there's some of these exotic species are going to be planted in some land cover types, probably more than others. So it'd be really, really good to understand that and also be able to like just filter out any error that comes through from like mistakes in the and where the points are being recorded. So we did that. We combine these with the host plant susceptibility ranking, which just comes from expert knowledge and also published evidence of how susceptible each of the species that we had in our model are. This ranges just from one, which is like gets infected but not very much infection, to five, which is like it just gets like absolutely covered in spores, and it's probably both high like is highly susceptible but also like really good at spreading myrtle rust to other species. So, from all these things combined, we come up with this local host density index, which is basically just what's the density of all the different hosts in New Zealand, where are they in New Zealand, and also just weighted by, well, how susceptible to myrtle rust is it? Because it might not be such a problem if you've got like loads of hosts that don't get myrtle rust much, but then a massive problem if you've got loads of hosts that get raging infections of myrtle rust all in the same location. We then combined this with the climatic predictors of myrtle rust risk that have come from the myrtle rust process model that Rob Beresford developed. We went for both sporulation risk and infection risk because I've been talking to them a lot, and we actually really like sporulation's really, really important as well, even though a lot of people currently just look at infection risk. And we finally combined these into our model and then calibrate it against the data because something I'd always been worried about as an empiricist moving into modeling is, am I just building some really convincing nonsense? So I felt it was really, really important to actually like calibrate and validate the model against the myrtle rust records. So we did that, and I think it's actually the first time when we publish this, will be the first time that someone's done that validation work. So yeah, big shout out to my colleagues at Plant and Food for helping with that. Anyway, we learned so much from this model. The first one that we just thought was really good to examine was, well, what land cover types actually do support the hosts? Because we wanted to look for crossover. And we're finding that kind of consistent with what we expected. It's the urban areas where there's both a lot of native species and there's a lot of exotic species that are being planted. And the worry is that they might actually be spreading the disease back and forth between them, especially when you've got highly susceptible species like lily pillies around. What was actually really interesting though is we actually have quite a lot of exotic species in regenerating forest types in mānuka and kānuka and in broadleaf indigenous hardwood, which is just a fancy way of saying like native scrub. So that's kind of concerning because like I've been wondering like could that be a bit of a stepping stone where, we built this kind of combination of exotic species and native species growing together? Are they building up kind of more disease and more spores in those areas than if, say, we didn't have any exotic species there? I'd love to know how this interacts with other land cover types. And we didn't look at it, but I think a really interesting question is like, what land cover types are next to other land cover types? And do we have, say, myrtle rust spilling over from urban parkland into broadleaf indigenous hardwoods and then onto indigenous forest? But that's a question for another time. The next question was, where are the exotic hosts? This actually was kind of contrary to my expectations. Turns out most species are restricted to the North Island, and Auckland City in particular, you can actually see it comes up red in most of these maps. It actually does seem to be a hot spot. A bit of a consideration that we're scratching our heads around is how much of this is reporting bias. There might just be a whole bunch of people in Auckland who go out like reporting every single lily pilly hedge because they're really into it and there's a big population. Um, that's not something that we can really account for, but we can certainly discuss in the questions. In comparison, the native hosts are pretty much spread across the country as you would expect, and I was really pleased to see that our results, even though we used a different model type, our results are fairly similar to the work of James McCarthy and his colleagues when they also did a species distribution model. So that was cool. When we put this together into our weighted host density index, we can see it's quite patchy, which is probably going to be quite limiting to, where myrtle rust can actually spread in New Zealand, because obviously you've got to have a host plant for the myrtle rust to grow on for it to be present in an area. But I think the key thing that we notice is that the local host density of the indigenous species is about 10 times higher than it is for all the exotic species combined in our model. And so I think the relative importance of the exotic host species is actually quite small compared to native species across the country. And I think that's just because they don't seem to be as abundant yet. So that is some good news. I'm showing you this. These are inputs, but I'm just showing you this because they're really important components of our disease intensity model. And you can see that both infection risk, that's the IR, both infection risk and sporulation risk, it's the highest in the wettest, warmest parts of the countries. And it actually gets low, in fact, very close to zero in cold, dry, and alpine areas. Keeping those patterns in middle rust infection sporulation risk in mind, we can now add in our host density index records. And if we do that, this is how we calculate our disease intensity index. And you can see the model form is disease intensity index equals sporulation risk times infection risk times the host density index. And then we've got this funny little number up here. So this is our scaling parameter, and we've used it to kind of understand the relative weight of the distribution of hosts versus the climatic parameters that affect myrtle rust infection risk. We calculate that. You can see it's here. It's the main thing to notice is it's patchier, like the levels of high risk are patchier in distribution than they are in either the sporulation risk or infection risk kind of submodels that feed into this. But we validated this index against the 2020 to 2023 middle rust records, and we did actually find that when we just include the disease intensity index model, it's the most parsimonious for explaining middle rust presence, both of in all of New Zealand, and also in the North Island only. So that was really, really good to see. So taken together with this scaling parameter of around zero point 0.5, we can kind of conclude that the climatic effects on middle rust epidemiology and host distribution matter, but hosts actually matter a bit less than the climatic variables that promote sporulation and infection risk. So sometimes what disease modelers will do is you can do validation in two ways. You can either do it quantitatively, like what we did in the last slide, or you can look at it qualitatively. We did both in this case, and it's basically just a matter of looking at your model outputs as well as the quantity that you're trying to predict, which in this case is the middle rust surveillance records. And we were pleased to see that they do match up fairly well with, most of the risk being most of the records being distributed where most of the risk is. And after that, we decided to try to kind of refine how do you interpret this model? Because the thing is, the disease intensity index is just a number, right? The actual numbers don't really matter that much. It's more about is this quantity higher or lower than the other one. So what we did here is we basically matched up the model outputs against the myrtle rust records and looked at quantiles. And basically what you can see here is we've created a category system, a little bit like the category warning system for hurricanes, where 9 is the highest and 0 is very, very low. And the key takeaway from this is these areas in the dark red, that's category 9. This is where, in this tiny, tiny little area, this is where 10% of all the myrtle rust records are in New Zealand. If you take category 9 and category 8 together, then this slightly bigger area contains 20% of all the myrtle rust records in New Zealand, and so on and so on. So you can kind of you can look at that and hopefully get a fairly like instantaneous picture of, oh yeah, that's category 9, that's dark red. There's a lot of myrtle rust. likely to happen in that area. And what's a really scary thing about this model is that you can see up in the top of the North Island, there's a really, really high risk up here. And this is actually where all the Bartlett's riders are. So they're one of our really rare and very beautiful myrtle species that people really love. And this is most of their natural home range. So this is not a good result to see. Finally, to answer our question, well, what's the impact of exotic species on disease intensity? So the 9 exotic species that we considered have the greatest local influence on disease intensity index. They're in the northern North Island, um, which is kind of good because at least they're restricted. Especially they are in Auckland City. And just to show you this, I know I shouldn't laugh, but I suppose at least it's a localized problem potentially. We can see this really, really, really high concentration of disease intensity, but also there is a fair amount of sort of disease impact that's potentially influenced by exotic species up around Whangarei. And also the Kaipara Peninsula as well. This is all eucalyptus salignina plantations that we can see. Well, sorry, not necessarily plantations, but eucalyptus salignina that's been planted. So that's quite interesting. There's a very negligible impact of exotics in the South Island. And in terms of which exotics, again, I think it's important to remember that the relative impact of these 9 exotic species that we modeled, it's actually quite small. So the kind of that delta DI value, that's just simply the difference in disease intensity index if we construct the model with all the species versus if we construct the model with one or all of the exotic species left out. I mean, it's the numbers are like the impact is quite small. So that's good. But nevertheless, out of these species, it's 4 of the lily pillies, so Syzygium australis, Syzygium floribundum, and Syzygium smithi, that have the highest local impacts on disease intensity. And I think looking at this, it's probably fair to say that these species should probably be controlled, or at least maybe just like banned from nursery sales in the warm parts of the northern North Island, because that seems to be where they're going to cause problems. And what did we learn from all this? Like heaps. So, our model might not be completely accurate, even though we're actually happy to see it's actually pretty accurate. And basically, we learnt that urban areas and regenerating native forests support both native and exotic host species. So I think there's some environments that we need to scrutinise in terms of how many hosts are there for myrtle rust and what are they potentially doing to the myrtle rust? What's sort of the abundance of myrtle rust that's around. Again, the combined impact of the 9 exotics studied, it's about a tenth of that of the native species. So they're abundant in some areas, but they're not as abundant as the native species. These 3 lily pillies have the highest local influence on disease intensity. And I think those species should probably be controlled, especially in Auckland City. Something that's really interesting is that we probably don't need to control exotic hosts of myrtle rust in the lower North Island, the South Island, or Rakiura under a current climate scenario. But climate change might change this as these areas get warmer. And so I'm in the process of just getting some climate change scenario models from Trevor Carey-Smith at NIWA. So we'll be able to actually model that and just see like, do we kind of need to be eradicating or controlling exotic species further south as the climate gets warmer? And another question that I want to answer with these models before we publish is what about all the planted Pōhutukawa outside of the natural range? Because you probably didn't see the numbers, but compared to all of the other native species, this Pōhutukawa has really, really, really high densities. And you can see it's in these 2 urban areas in Wellington and Auckland. And this is something I actually believe, and I don't think it's an artifact of the model because I was in the centre of town the other day and I took this photo and you can see 1, 2, 3, 4, 5, 6, 7, 8 Pōhutukawa just planted in that like probably 50-metre strip of street. And I mean, I think I counted 50 along the bus route on the way to university, and that's only a half-hour bus ride as well. So they are beautiful. They are a taonga. But I think it might be sensible to just have a think about, at least for us to model what are these really high densities potentially going to do with myrtle rust risk, especially when they've been planted north, like south of their natural range, which is the northern North Island. So we learned heaps. And I was actually just gonna give a shout out to all the other women who are having babies. I did a lot of this stuff while I was on maternity leave, and I just wanted you to all know it is possible. This is how it looks. Um, your cat and your husband suggesting that you should go to bed, and it gets done at night with the baby monitor. So I think it is possible to do it all, and it's been a lovely experience too. And more seriously, I have so many wonderful people that have been involved in this project that I need to thank. They are all in this list and have either co-authors or they're people who have allowed me to bother them a lot. So just want to say thanks. And that's me.
All my te paki virtual claps and love hearts coming to you and your amazing kōrero, Sarah. Thank you. I was, have to admit, I was laughing very hard for some of your talk, laughing in a good way. For example, I've never heard someone say um, to me that essentially all models are wrong, but some are useful.
Oh, it's actually a really famous quote by a statistician. I forget his name is G Box. He's the guy who invented box Cox
Oh.
models. But yeah, it's actually become a core tenet of statistical modeling, and I think it's great.
Well, I think we can say that your model is useful and you've done some validation and work with people. So even if yours model is essentially wrong, it is useful. So I like to
Thanks.
say, um, we're gonna, for those of you who want to ask a question, please put it in the chat and I'll read it out. We've already got one there. I just also wanted to thank you for giving such a sort of a upbeat kōrero around what is essentially a rather downer topic. I have to admit, I'm one of those people that walked around with Emma Bodley from the Auckland Botanic Gardens at the time of the response, looking at myrtle rust on lily pilly hedges in the very, how would you say, highly wealthy suburbs of Remuera, Howick, and other places. St Kentigern's the very wealthy girls' school. Its hedges were absolutely covered with it, and we were watching the school kids and others like brushing past it, and plumes of spores, Deridia spores, were going into the air, and we could literally see it. But that was when the response was just about to be stepped down, and we thought, well, yeah, that's going to be stepped down. So I'm sorry, I'm one of those that contributed to those lily pilly hedge results. But
No.
I have to say, it is a in the urban spot a terrible inoculum source. Um, One thing though is that it is on the monkey apple,
Mm.
the what's it called, Syzygium
Oh,
smithii.
Jambots.
Yeah, that has is a national under the what is it called? The National Pest Planter Corps, which MPI oversees,
Mm.
and the Regional Pest Management Plan, which is our Auckland Council's one. Although it's grandparented, i.e., if you were one of those hedge owners in Remuera, you don't have to do anything. But they have put in restrictions around selling it and propagating it and moving it even more. So that's at least in place. But then I suppose, is that being monitored? And is Auckland the only place that needs those restrictions? So that's just a couple of comments from me. But you've definitely, what your mahi has done is shown some key inoculum sources for and those risk sites in Whangarei and Auckland. There are actually really rare taonga in those regions that we do want to protect. So kia
Yeah.
ora for your work to point that out. So yes, please send your questions in. Kia ora, Sam. We've got one here from Sam. Kia ora, welcome, and thank you for joining us. He's interested in the relationship between the resilience of Rākau and the health of the ecosystems and the complex biodiversity. Most of Māori Tāwaka populations are only just holding on and are growing in isolated situations, which I think you mentioned. So wondering if the niche ecosystem were thriving and the Māori was strong, would that mean that the rākau would be more resilient to infection and disease? Thank you, Sam. That's an awesome question.
That is an awesome question and a really complicated one. But I'm glad you asked it. So based on a lot of conversations with Ra Smith, we we actually came up with a proposal to try to look at the interactions amongst the rako in wetlands. It would have been three key species, Maritawake, Pukatea, and Kahikatea, because we really wanted to know a couple of things. Like we wanted to know like if myrtle rust just destroys all the Maritawake, like what's going to happen to the other species that are there? Like will they come and fill their place, or is this going to leave like a gap in the ecosystem that's most probably going to get filled by willows. Unfortunately, we applied for two Manatua Papa fellowships at a Marsden, and we haven't been lucky with any of them. So I'm thinking about what to maybe do with that project. Maybe that's something we hand over to a PhD student because it's all written up and all budgeted. So we've done that work there. Um, in terms of, oh man, I don't know if Hannah is here. That should be a really good person in the audience to help with this. I don't know how the health of the surrounding trees would impact Mighty Tawaki. My suspicion, based on what we do know about biodiversity impacts, is generally speaking, more biodiverse systems where trees are thriving are harder for invaders to get into. But that's not really for disease. So I think that would be a really, really, really good question for someone to go and research. And I think it's research that sorely needs to be done. So ka pai.
Yeah, kia ora, Sam. You mentioned a really good point about Hanarea e her tamaunu, who's on maternity leave as it happens,
Oh,
but she presented her Rangahau Rising kōrero last year in July around the microbiome and how that is affecting how on Maire Taiwaka and how the myrtle rust is affecting I guess, the natural population of the good beneficial parts of the microbiome. And I urge people all of the rangahodizing presentations, including today's, are actually online available and for you to look at, including last year's one, which Hanarea's
mm,
presentation is part of. So just we should actually go back and look at that at Hanarea's one, but she presented some very clear results on that line. And if it's affecting the natural microbiome, that's not good. That's another negative impact. And also
mm,
to add on to your question, Sam, around looking at those places that the rākau are actually hanging on, as you say, in really small pockets. It is it's a really terrible situation for these rākau and like conservation efforts by iwi, by landowners, by the councils and by DOC really focusing in those areas. I think that's something your model can assist with. Where are the areas that should be focused on first? But kia ora,
mm.
Sam, that's a fantastic question and yeah, one that definitely needs more answers. Um, Kia ora, Dave Haval. Oh, no, my haramai. Have you checked the disease intensity in the field against the model, especially in Wellington?
No, and I'd love to. Um, yeah, this is something that'd be really good. It's quite hard and actually quite expensive to get a measure of disease intensity for myrtle rust, but there's some really interesting work that's being pioneered at Australian National University where they've got these like really big like filtering machines that effectively suck in the spores from the atmosphere and can give you an idea of what the spore load is in a particular location. The only problem is they cost $10,000 each. Um, but, I mean, there are other ways to do it as well. That would be the best way. But I actually think something really practical that everybody can do is record your Myrtle Rust observations in the Myrtle Rust Reporter Project in iNaturalist because something that doesn't cost us money and something that, I can go back and do later on is I can keep pulling down those records from iNaturalist and feeding them back into calibrating the model. So over time, we can actually like I think the best models work when you've got a model that then kind of feeds into a field study that then feeds back into refining the model, and they work in harmony, not in isolation. So yeah, so please record your observations.
Yeah, kia ora, Dave. I think I want to tautoko your whakaaro, Sarah, to say there's a lot more mahi and work and research to be done. And it's sort of, we've come again at the end of a natural cycle with the mahi that Beyond Myrtle Rust was doing, and there was some in Ngārako Taketake, and there was been, those two programs have finished naturally. But as you say, there's been the mahi done here in Bioprotection Aotearoa, which is explored some key pivotal areas and answered some very good knowledge gaps. So kia ora, BA, that's great what's happened here. But also I do know that there's been some, as you said, some funding applications gone through, and there's been very few, if any at all, that have, you know, been granted for the likes of yourself and others to continue some of those key areas. So we really do need to have a sort of some funding to continue in this area, just because in my observation, plus what I one of my colleagues, Becky Ganley, has been observing, and a number of other of you have also, has been seeing an explosion of infection and some very bad symptomatic diseased areas this year, and particularly the very warm, wet summer we've had in parts of Aotearoa. And maybe we should open the floor really to actually ask you all who are there, what have you also been observing in this season? Because I, for one, have seen more, which I hadn't compared to the previous year, which was a lot drier. And then also Becky had also seen this. I'm wondering who else has observed this as well. So I guess that's a collective
Mm.
shout out to the people here, and you put in your comments in the chat or another question. But it's something that we're watching this whole incursion develop and spread and behave in a new environment, which is that of Aotearoa. It's a key critical time to actually follow a disease's incursion and where it leads to and what it's doing. And so now, this is a plea. This is now is the time to be doing the research and doing the monitoring and doing the observation. So I think it's awesome that, as you say, Sarah, keep using iNaturalist.
Mm.
Anything else you'd recommend?
Oof. I mean, apart from doing systematic surveys, I think the most feasible option right now is just to keep reporting those observations.
Yeah. Dave's commented here. Thanks, Dave. Haven't seen much, mostly been in Paraparaumu, which has been dry. Yeah. I
Mm.
think I think some of the observations Becky and I were talking about have been like in Whangarei and places like that, which have quite a warm, wet summer.
Mm. I've been keeping an eye out for it in Wellington, and actually haven't seen any this year, which has been quite interesting. But certainly it was when we were in the field for Pearl and Teddy's projects. That was autumn 2023. We were seeing it on a lot of the Māori tawaki, particularly the seedlings. No, I'm gonna take that back. It was on both. It was both adult trees and seedlings. Um,
Yeah, that's the other thing is to often remind people don't just look on the canopy, or don't just look on the leaves. Look at if there are seedlings, that's a part of the life cycle that often gets of a susceptible plant that gets absolutely hammered. So that is one that people could look at. Also, trying to get up into the canopy of some rako, that's an interesting scenario, unless you've got binoculars or something. But looking at the seedlings or the young, if there is sort of regeneration in the area, that is one key area to look at as well.
yeah, and actually with Christy Stevenson's work in Australia, they're finding that the composition of the seedlings is what's being affected first, because obviously it takes the older trees a couple of years to die. And in terms of just like population, metapopulation biology, that might end up being the pinch point for kind of the long-term viability of some of these tree populations.
I got a couple of observations from Wairarapa. Uh, yes, I mean we've seen a trend of probably a series of wet, warm summers. It's the weather patterns are changing. A lot more easterly systems bringing in the rain, and quite interested in the vectors that not just myrtle rust but other pest species bring, in particular like pest plants along the rivers, but then the windborne um, diseases and species seem, and I'm wondering, like, because the prevailing wind is northwest in the Wairarapa, and the spores from the myrtle rust traveling. Well, this is what Hanarea was saying, is the spores are sort of traveling on the wind
Mm.
and hitting populations or plants, myrtle plants, in the path. And so I'm wondering climate change, what role climate change will take. Other things I've noticed since talking about this myrtle rust species is I've been noticing a lot more mauri tawake, been finding new, well, I'm not sure if they've been surveys, but new spaces with um, mauri tawake, kiri whakapapa, northern Wairarapa, which has been like, wow, oh, um,
Oh, yeah.
yeah. So we really cool to show you those trees, but it's typical. It's surrounded by open paddocks, monoculture, like sheep and beef farming, and they're kind of like small stands that sheep and cows take shelter under. And so to find, and also there's deer and possums, which is the other big
Mm.
um, disruptor in the system, is those
Mm.
existing pest animals hoovering up all of the seedlings and
Oh,
seed,
cool. Yeah.
um,
Oh,
which is another reason why you don't see them so much. But I couldn't see any. I couldn't see any myrtle rust on the trees that I saw, although they were quite big, old, gnarly ones. Um, so difficult to climb and check out the leaves. And
That's
I'm
interesting.
no
So I'm
rich.
actually I'm staring at the model now actually to just see what the risk is like up in that area. You can't, maybe I should screen share again and show you.
Yeah, the main reason, the main place that we had seen infected was Lowe's Bush.
Oh yeah.
Yeah,
Yeah.
Allen's Bush, that right in the middle of open plains, so a little island
Mhm.
in a desert of farmland. So the stands that I'm talking about were more in the foothills of the Tararua,
Oh
quite
yes.
sheltered and kind of small, kind of yeah, niche spaces, I suppose.
Yeah, yeah. Actually, the foothills of the Tararuas, that's actually where the disease risk, well, it's when bottled disease risk is lowest. So we kind of suspect, I didn't talk about it, but what we suspect is, yeah, that area might be a really good refuge for Māori Tawaki. And
Mm,
I reckon there's probably quite a few more trees kicking around the area as well. Because like, they don't seem very dense. Like, we found some up one of the tracks around, on the way to Holdsworth.
yeah,
And there's just a couple, but none of them had myrtle rust. It was really interesting. And I think it's a bit colder and they don't seem to be high density. So I think
yeah,
maybe those two things could interact
yeah, yeah,
and just
yeah.
make it a bit of a safe haven. So it'd be really cool if we can get more trees planted into those sites.
Mm, mm,
yeah. Oh, well,
yeah.
no, kia ora. Well, let's keep that kōrero going.
Great work,
Especially,
Sarah. That's really cool. And thank you very much for your kōrero. It's really inspiring.
Oh, thanks. Yeah, it'd be good to keep in touch.
Absolutely.
Yeah, no, that's awesome. Kia ora. Thanks, Sam, for that joining in and welcome again and thank you for your time. And also, again, a big homai te pakipaki to Sarah and her amazing talk. I loved it. And we're just reminding you that for those that missed and want to watch it again, it'll be online in the Rangahau Rising, part of the Bioprotection Aotearoa website. And also, if you want to, and please do, register for next month's Rangahau Rising speaker. It's going to be about one of our other nārara pest problems and with the rākau Cody dieback. And we're hearing from Dr. Mariana Tararo on understanding the key weapons of Cody dieback. So that's on the 17th of June next month. So please register on the Rangahau Rising part of the website so you can join us for that kōrero as well. There's the link. So also, thanks Sarah and Zohar for helping us bring this to you today. And again, thank you, Sarah, and all of you for joining us. Thank you. I'll close our hui with a karakia whakamutunga and then bless us on your way for today. E te papatua nuku kua mutu mātou e te tahe o te rā, kia tātou rangamārie kia mātou. Mauri ora. Ka kite
Mauri
anō,
ora.
everyone. Take care. See you next month.